Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Okay, hello, welcome back. Sorry I've been a bit busy, I've had exams and stuff. Welcome back to the A-level cookbook. Today we're going to be doing paper 3 from June 2019 for biology. So it says, describe how ultrafiltration occurs in a glomerulus. So you need to remember the structure of your kidneys and stuff. So just as a quick reminder of what the kidneys look like. So inside the kidneys, you've got loads and loads and loads of filtering units. You've got your afferent arteriole here. You've got this big bundle of capillaries here and an efferent arteriole that comes out. And then you've got your Bowman's capsule here and so on, right? But they're only talking about ultrafiltration. So we don't need to worry about the rest of this for now. That's all about selective reabsorption. So what happens is your afferent arteriole is remember arterioles come from arteries, so it's got quite a high pressure in here, and the efferent one's actually a lot narrower, so it's actually quite much smaller. So you have this relatively high pressure here and low pressure here. So imagine you've got like a hose pipe and you squeeze the end of the hose pipe. The water comes out way faster, it's squished out. So that's kind of what happens here is that this is the narrowing of the hose pipe, and what happens is all the stuff is squeezed out little pores like this. So that's what the process is. So you would write a high blood pressure causes substances to be squeezed out of the glomerulus into the Bowman's capsule through the basement membrane. This is just a general answer I'd recommend saying. And in this case, you should say what the substances are. Just generally speaking, with all questions, when you say substances, you need to say something. So I'm going to say water and ions. Just generally speaking, the mark scheme here says you need to do two, but there you go. So glucose and water are reabsorbed by the proximal convoluted tubule of a nephron. Put a tick in the box next to the correct ways in which glucose and water are absorbed. So glucose by active transport and water against a water potential gradient. No. Glucose by diffusion and water down a water potential gradient. So the down a potential gradient, yes. Diffusion, no. Glucose by facilitated diffusion and active transport and water against a water potential gradient, yes. Oh my god. So in the proximal convoluted tubule, so you've got your Bowman's capsule, whatever. Convoluted just means wiggly. So you've got this proximal convoluted tubule. The goal of the nephrons is to try and absorb as much glucose and useful stuff as possible. And generally speaking, glucose, nearly all of it's absorbed in normal individuals who aren't affected by kidney disease and stuff. So the proximal convoluted tubule is going to use facilitated diffusion, but there might come a point where there's more glucose on the outside than in. So what it will also use is active transport to make sure that every single bit is absorbed. So the equation shows the relationship between urine concentration in arbitrary units y and the mean length of the loop of Henle in millimeters x. Calculate the mean length of the loop of Henle in an organism that produces urine with a concentration of 15.56 arbitrary units. Whenever you have a calculation question, if you can't do it within a minute or however many marks it is, mark a minute, just leave it. There's no point doing it. It's so much work for nothing. So it says that 16.56, that's going to be 16.56 equals 0.72x plus 4. Take away 4 from both sides, so 0.72x equals 12.56. Divide 12.56 by 0.72. So 12.56 divided by 0.72 gives you 17.44444, and yeah, 17.4 millimeters. Scientists investigated the relationship between the thickness of the kidney medulla of different species of mammals and the concentration of their urine, and figure 1 shows their results. So you've got the concentration of urine on this axis, and you've got the thickness of the medulla, and you can see there's a roughly speaking, as the thickness increases, the concentration increases. So the question says, explain the pattern showed by the results in figure 1. So not describe, don't say as one goes up, the other one goes up. You need to explain why that happens. We can see that as the thickness increases, the concentration of the urine increases as well. So we need to think about what's going on here. So the first thing you need to appreciate is that if you've got a thicker medulla, you have a longer loop of Henle. These questions nearly always test that sort of fact, that this organism has a longer loop of Henle. So thicker medulla gives you a longer. So we need to now explain sort of the implications of what a longer loop of Henle means. So what happens is So what happens is near the top of the ascending limb of the loop of Henle, sodium ions are pumped out into the medulla. And generally speaking, where sodium goes, water follows. Because if you think about it, water. So if I draw like an example, so let's say we've got the ascending limb here. Sodium moves out here. Sodium ion, sorry, that's going to decrease the water potential, so water is going to follow as well. So if you've got this longer loop of Henle, you've got way more sodium coming out, an increase in sodium concentration in the medulla, so more water follows. So therefore, there's an increased sodium ion concentration in the medulla. So the water potential gradient is lower. So more water is reabsorbed by osmosis from the loop of Henle and collecting ducts. Describe the role of saprobionts in the nitrogen cycle. So saprobionts, what they do is they return nutrients to a cycle. So things that animals don't eat, like poop, bones, this and that, blah, blah, blah. With these kinds of questions, you can't just say they release substances from waste products. You need to say what they actually break down and what the useful substance is. So what they do is they use enzymes to hydrolyze, and you could pick whatever. So proteins, DNA, urea, whatever. So they hydrolyze proteins in dead or waste material. And what does this do? Well, this releases useful substances. So this releases useful substances. And you need to say what the substance is. In this case, amino acids. With these kinds of questions, people always slip up by saying they break down dead or decaying material, and they don't say what the actual substances is. So just generally speaking, if you ever have a question on saprobionts and stuff like that, just say they, break down dead or decaying, dead or waste material, and they break down proteins, releasing ammonia, ammonium ions, amino acids, and so on. One environmental issue arising from the use of fertilizers is eutrophication. Eutrophication can cause water to become cloudy. You were given samples of water from 3 different rivers. Describe how you would obtain a quantitative measurement of their cloudiness. Quantitative means you can put numbers to it. You can measure it with numbers. Qualitative is like a descriptive term. So, for example, kind of cloudy, not very cloudy, very cloudy. So we need to try and think of a way we can measure cloudiness and put it numbers with it. So although you haven't done a specific practical with this, you should be able to sort of derive a practical from this. You've got differing levels of cloudiness in stuff. So you've got that, and you've got this, and then you've got like a very clear one there. Cloudiness happens because light can't get through as very well. So in this one, like, some light gets blocked and reflected and this and that, blah, blah, blah. This one, not very much light comes through. This one, all the light comes through. So we need to think of a way to be able to measure the amount of light coming through and put numbers to it. And that would be using a colorimeter. So use a colorimeter on each sample. You measure the absorbance. You need to tell them what you're actually measuring. Absorbance. And then you just plot on a graph with different things. But you need to discuss how you would standardize the method as well. So for example, if this has got lots and lots like dirt and dust in it, it might settle towards the bottom. So you want to make sure it's consistently throughout. So you would ideally shake the sample before testing. Or other things you can talk about is using the same volume of water, making sure you calibrate the colorimeter, so setting it to 0 as well, or using the same wavelength of lights. In this question, a calibration curve isn't really relevant because they're only asking just for a number of how cloudy they are. So they could use a very clean supply of water, something that's filthy and something that's in between, and be able to put numbers to it and sort of work from that. So in this case, you just need to talk about how you use a colorimeter and shaking the sample before testing and zeroing as well is or something I would always add. The colorimeter, sorry if I said colorimeter. Right, so figure 2 shows a photograph of a dissected heart. So they've labeled the left atrium, they've got valve A and chamber B here. So it says name valve A and chamber B. So valve A is the left atrioventricular valve. So left atrioventricular valve. also known as the mitral valve or the bicuspid valve. And then chamber B is the left ventricle. Okay, so generally speaking, the heart is mirrored. So usually the right is here and the left is here when you're looking at diagrams of it. But in this case, they've put the left on the left and the right on the right. So I'm just going to follow their diagram. So your heart has the left atrium, you've got your left ventricle, you've got your right atrium, and you've got your right ventricle. In the right, you have the superior vena cava and the inferior vena cava, which both drain into the right atrium. They bring deoxygenated blood, right? So then what happens is the right atrium and left atria contract and it shoves blood into the right ventricle. So you have to go through valves and they're between the atrium and the ventricles. It's going through the right atrioventricular valve. At the same time, the left atrium is pumping oxygenated blood into the left ventricle. So that's going to be through the left atrioventricular valve, also known as the mitral valve or the bicuspid valve, and this is also known as the tricuspid valve. Then what happens is the ventricles contract, and it depends on where you want the blood to go. So this is deoxygenated blood. So we want it to go to the lungs to be able to become oxygenated. So when the right ventricle contracts, sends blood through the right semilunar valve, also known as the pulmonary valve, and that goes into the pulmonary artery, and then that goes to the lungs. So it's the pulmonary artery. And then with the left ventricle contracts and that goes to the rest of the body through the aorta. So that's going to be the aortic valve. So bang, that goes to aorta. Okay, so it says give 2 safety precautions when dissecting a heart. It says ignore goggles and very basic things like take care with scissors. Because let's be real here, there's a pretty obvious things. So I would say something like wear gloves or wash hands or something like that and cut away from self or from others, or cut on a hard surface, disinfect equipment, stuff like that. It's specific to dissecting a heart. Wearing goggles, you're doing pretty much anything that involves any sort of liquid, and taking care of yourself or keeping sharps away from fingers is something you would do with all cases of sharps. It's asking for more specific things to do with dissecting a heart. Okay, so it says explain how valve A in figure 2 maintains a unidirectional blood flow. So valves generally work by sort of like this, and the pressure behind them has to be higher than in front of them. If the pressure is higher, then what it does is it shoves the valves open like that and blood's ready to go. If the pressure isn't as high, then what happens is the valve stays shut. So if the pressure after the valve is higher than before, it stays shut. So you need to make sure the pressure is high enough for that to be the case. So it says explain how it maintains unidirectional flow. Well, the valve opens if pressure in the left atrium, I'm obviously abbreviating, but you need to say that, is higher than in the left ventricle. And that makes sense because when the left atrium's contracting, you want to get blood into the ventricle. You don't want it to go back up. But then the left ventricle needs to pump and get everything going around the body, but we don't want it to just shove blood straight back into the atrium. So the valve closes if the pressure in the left ventricle is higher than the left atrium. And that allows unidirectional flow because, again, I'm going to draw a better diagram here, sorry. So we've got our valves here, we've got a left atrium, we've got a left ventricle. If the left atrium's contracting, this one's going to be relaxed, so the pressure's higher in the left atrium, so bang, the blood goes through. Whereas if the left ventricle's contracting, the pressure's higher in the left ventricle than it is in the left atrium, so it pushes back on the valves and shuts them. Like it's a one-way system, it can only open through one way. So a research scientist investigated the effect of caffeine on heart rate in human volunteers. The scientists divided volunteers into 3 groups. Each group was given the same volume of fluid. So each group of I was given a sports drink containing caffeine and sugar. Each member of group J was given a sports drink containing caffeine and no sugar. And each member of group K was given water. The scientists recorded the volunteer's heart rate before the drink was given and for 120 minutes after the drink was given, and her results are seen here. So we're measuring the mean change in heart rate. So if the mean change is 0, there's no change in heart rate. So therefore it stays the same. If the change is negative, then it means their heart rate decreased, and if it's positive, the heart rate increased. And this is where we have the time after the drink was given. So group I is the straight line, group J is the dotted line, and group K is the dashed line. So you can see in group K, the heart rate kind of went up and then went back down. In group J, what happened is the heart rate kind of dropped and then started climbing, and in group I, it kind of dropped and started climbing as well. So caffeine affects the autonomic nervous system. Suggest how caffeine could account for the results in I at 60 minutes. So let's look at 60 minutes then. So what happened with I? Oh, so in I, the mean change in heart rate was about like what, 7.5-ish? So that means their heart rate increased. So one mistake that a lot of people I see make in any nervous question is they write signals or information or messages. Those are just you come on, we're doing A levels now. Don't say that. And I have an interest in neurology as well. And just saying information is just don't say information, okay? Just do not say signals. Just say the word impulses, please. So what happens is the thing that controls heart rate, so the heart beats on its own, it's myogenic, but the thing that controls the impulses is the sinoatrial node, which can be influenced by things. So your heart can beat on its own because the sinoatrial node does the job, but something can tell the sinoatrial node, hey, beat faster. So in this case, what's happening is the sinoatrial node is increasing the heart rate, so we need to think about why that happens. Well, the two things that affect the sinoatrial node are the sympathetic and the parasympathetic nervous systems. So got the parasympathetic nervous system, the sympathetic nervous system, and you've got your sinoatrial node, and then you've got your heart rate. Right. So sympathetic, fight or flight. So you're gonna want to increase heart rate. Parasympathetic, rest and digest. So you want it to, calm down. So in this case, the person's heart rate is increased. It's probably going to be the sympathetic nervous system that's doing the job. So now we need to talk about how we actually write this down. So what happens is that there are more impulses sent via the sympathetic nervous system to the SAN, and this leads to an increased heart rate. Please do not say information. Please don't say signals. The idea here is that there are more action potentials being sent. If you have more action potentials, then you have an increase of that information being perceived, if that makes sense. The intensity of a nervous impulse depends on how many action potentials you send. Before taking the drink, the mean heart rate of J was 68 beats per minute. 15 minutes after taking the drink, the mean volume of blood leaving the hearts of group J was 4,700 centimeters cubed per minute. Calculate the mean volume of blood leaving the heart at each beat 15 minutes after taking the drink. Each beat 15 minutes after taking the drink. So at the start, before they even did anything, their heart rate was 68 beats per minute. We need to work out what their heart rate was 15 minutes after. So after 15 minutes, their heart rate was around here, which if you read across the scale is a mean change of -4. So 15 minutes later, their heart rate was actually 64 beats per minute. So what this means is there's 64 beats in 1 minute, and each minute they're shoving out 4,700 centimeters cubed of blood in 1 minute. So that means that 64 beats is shoving out this much blood. So one beat would be 4,700 divided by 64, which gives you 73.4. Increase in group I could be due to the combination of caffeine and sugar. Suggest one drink to be given to an additional group that could be investigated to find out if this is true, and give a reason for your answer. So what they've done is they've got a caffeine and no sugar option and caffeine and sugar to see if there's any differences between adding sugar when you already have caffeine. And then they have another group where there's just water. So they're trying to see if caffeine has a role on this. If we wanted to find out if sugar had a role, then they would have to add another drink where it's just water and sugar with no caffeine, because we don't want caffeine to potentially affect the results. So the group should be given sugar, water, and no caffeine. You need to say no caffeine. You need to make that like crystal clear. Why? To see if the increased heart rate is due to caffeine alone, or if sugar plays a role. Mitochondrial DNA, mtDNA, is small circular DNA found in mitochondria. It is 16,569 nucleotides long and contains 37 genes in a control region. Sports scientists investigate whether a mutation in the control region of mtDNA in human males is related to the ability of being able to exercise for longer. So the males in group T had a thymine at nucleotide position 16,519. The males in group C had a mutation resulting in cytosine instead. So the control regions of T and C were the same length. Describe the type of mutation that's likely to have occurred. So you have the same length, but the base there is different. So it has to be substitution. Okay. Group T and Group C completed the same 8-week training program. The following measurements were taken at the start of the 8-week program and then again at the end. So they got VO2 max, which is a measure of maximal oxygen uptake, and then they've got citrate synthesis activity. So CS is an enzyme involved in the Krebs cycle. The scientists then calculated the percentage increase in each measurement in both groups. So Group T, they've got a bigger percentage increase in VO2 max compared to Group C, but both of them still had an increase in VO2 max. Okay, percentage increase, if it's a 0, it means there's no change. If it was negative, it means it decreased. So both of them still had an increase. The increase is higher in Group T. Same goes with CS as well. They both still had an increase because none of them are 0 and none of them are negative, so there's no decrease. It's just Group T has a bigger increase. So a student concluded from Figure 4 and Figure 5 that training has a positive effect on VO2 max and CS activity. Well, like I just said, both of them had an increase, so that does support it. Whenever you're seeing an evaluate question, you need to say what supports it, what goes against it, and what other things are missing. Statistical tests, are there other factors involved? Timelines, ages of participants, sex, whether it was done in humans, whether it was done in a test tube, whether it was done in a petri dish, whether it was done in a body, loads of different things like that. So all they're saying is that doing training has a positive effect on VO2 max and CS activity, not exercise as a whole, just these two factors. And to be fair, the results show that. So first of all is that VO2 max and CS activity increased following, for both groups. So that supports what they said. However, they're saying that if you do that training, that works. And if you do training, you're not going to stick to doing 8 weeks. You're going to do way more. So it was only 8 weeks they did it for. We don't know after 6 weeks if it's just if it's sort of like stops or just levels out. As well as that, we don't know what training means or what training involves. It depends on what exercise it might be. So it might not be true for all exercises. As well as that, no statistical test has been done. No statistical test done, but you need to say why that's important. I've seen so many people that drives me bloody insane when they say, "Oh, there's no statistical test." Okay, so why do we need to do a statistical test? So there's no statistical test, so we don't know if the differences in each group are significant. You cannot say there's no statistical test and just leave it there. You need to say there's no statistical test, so, and then say what the so part is. So the differences could be due to chance, or we don't know if the differences are significant. Because statistical tests check correlation, they can check differences between two groups, and they can also check, differences between expected and observed results. We are comparing two different groups and two different factors, so we're looking to see if there's a difference between group T and group C, which would involve using the t-test. The mitochondrial DNA region is an area of mtDNA that's non-coding. This region stimulates the synthesis of both mtDNA and mitochondrial messenger RNA. Use this information to suggest 2 reasons why a mutation at nucleotide position 16,519 could lead to the differences seen in Figure 5. So, just as a quick reminder, Group C had a mutation where it has cytosine instead of thymine. And it says here that this region is non-coding, but it stimulates the synthesis of mtDNA and mitochondrial messenger RNA. So what might happen is you've got less transcription to make mitochondrial RNA and less translation into proteins. So we've talked about mitochondrial messenger RNA. Messenger RNA is involved in transcription and translation, making proteins, right? We need to talk about why mtDNA is relevant. Why would we want to replicate DNA? You replicate DNA in cells to replicate cells, so it's probably a similar thing where mitochondria replicate. So if you've got less mtDNA replication, then you've got less mitochondrial replication. This question's testing your knowledge of what the hell things do, basically. Messenger RNA is going to be involved in transcription and translation. It's going to be involved in making proteins. Mitochondria and everything in your body needs proteins, to work, like enzymes and structural stuff. So if there's a difference in the amount made, there's obviously going to be less protein, so the mitochondria is not going to be able to do as much work. It's not going to respire as well. Now, the other thing it's asking is about mitochondrial DNA. So you need to think, okay, when do we replicate DNA? What's the relevance of that? We replicate DNA in cell division. Therefore, it has to be something to do with mitochondrial replication as well. Right, a sports scientist investigated whether there is a correlation between the percentage change in VO2 max and the percentage change in CS activity in group T. So we're looking at correlation. Figure 6 shows their results. They've got percentage change in mass, we've got percentage change in CS activity. It says, well, I mean, if you look at this, there's not really a straight line pattern here. There's not an up, there's not a down. There's not really any correlation, to be honest. The results are just sort of sprawled everywhere. So there's not really any correlation. So it says having thymine at nucleotide position 16,519 in group T causes an increase in ability to exercise for longer. Evaluate this conclusion, use all the data in the question. So obviously we need to do the same thing as always. We need to say for and against. So let's look at for. So if we look at the first data from this previous question, where's it gone? Here, you can see that group T has increased activity in VO2 and CS activity, both of them. So remember that VO2 max is oxygen uptake. So if you've got increased VO2, you're taking in more oxygen for respiration. And CS is involved in the Krebs cycle. So if you've got more CS activity, you've got more of the Krebs cycle going on, so you have more respiration. So if you have more respiration, it means you can exercise for longer because you're making more ATP. So figure 5 shows increased CS activity in group T, therefore more, respiration. Figure 4, increased VO2, so there's more, increased O2 uptake, so there's more respiration. However, there's no correlation between the percentage change in VO2 max and CS activity. So what that might imply is that these two are not the only measures that are involved in sort of respiration and activity for longer. There might be other factors involved. Other things you can mention are that, that they didn't really exercise for very long. They did 8 minutes. Like, that's not really that useful. Okay, so the crown of thorns starfish cuts is one of the main causes of the decline of the world's coral reef. Marine biologists used a choice chamber to investigate the effects of flashing and constant light on the behavior of carps. Table 1 shows their results as they presented them. The p-values show the results of a statistical test. So we've got behavior of carts. We've got carts moving towards the stimulus, carts moving away from the stimulus, type of light used in the number. So 22, 28, 12, 38, and a p-value of 0.69 and 0.02. So it says state and null hypothesis the marine biologists tested in this investigation. So you should be able to conclude that we're going to be doing this is a chi-squared scenario here. We're expecting it to be equal between both groups. There's no difference. However, you can see there's a difference, and that's what the p-values are. So your null hypothesis has to say there's no difference between, what we expect or what we observe. But you can't just say there's no difference between expected and observed. That's too vague. It's too general, and the mark scheme says no. So we need to relate that to this example here. So in this case, it would be the type of light has no difference in the movement behavior parts. You could also say something like there's no difference in the amount of behavior and movement with a constant or flashing light. But that's what this question's asking. The natural habitat of carps as coral reefs of tropical oceans suggests two factors that should be kept constant in the choice chambers so that carps display normal behavior. So it would be things like temperature, pH of water, oxygen concentration, light intensity, I guess, salt concentration as well, distribution of food. A journalist studying Table 1 suggested that either type of light could be used to cause carps to move away from coral reefs. Evaluate their conclusion. So, suggested that either type of light could be used to cause carts to move away. Now, I thought this meant initially using a light somewhere else and dragging them away from there, but it actually means putting the light in there and getting them to basically move away. So if we look here, in flashing, 28 out of 50 move away from the stimulus and 30 out of 50 move away from the stimulus. So it's higher in both. So again, it says evaluate, so you need to say things that support. So there's movement away from the light in both cases. However, there's a p-value here, so you got to talk about that. So the p-value of constant is 0.02, so it's less than 5%. So p-value in constant is less than 5%. So there is significant movement away from light. The differences between the towards and away are statistically significant and not due to chance. However, in the other one, where you've got flashing lights, the P in flashing lights is greater than 5%. So the movement away is likely due to chance. What this is trying to get at is that you can see here there's more moving away than more moving towards, but it's statistically significant. It's not due to chance. There's probably something driving that. Whereas here, there's still more moving away, but more than more moving towards, but it's not statistically significant. It's probably that they got lucky with that result due to chance. So one of the reasons carts can destroy coral reefs in a short amount of time is because they move quickly, allowing them to move from one reef to another. Table 2 shows the maximum speeds recorded of carts in constant light. So calculate the shortest time a cart would take to move up a coral reef from 66 meters underwater to 18 in hours of daylight. So again, if you can't do a calculation, just leave it. So it says calculate the shortest time to move from a coral reef from underwater to hours of daylight. So they're moving towards constant light. So your speed is going to be 259 millimeters per minute. The distance they want us to travel is 66 minus 18 meters, which is 48 meters. You need to convert that to millimeters. So it's going to be 48,000 millimeters. So time equals distance over speed, which is going to be 48,000 over 259, which gives you 185 minutes. So then you divide that by 60 and you get roughly 3. It's about 3 hours. Uncontrolled cell division can cause tumors to form. Figure 7 shows the growth pattern followed by a type of tumor. So we've got time in years, we've got log 10 to the number of cells, and we've got the size of tumor that can be detected. So anything above this line can be detected. Fair enough. Use figure 7 to calculate the percentage of maximum growth in this type of tumor before it can be detected. You will need to use the 10 to the x button on your calculator. Okay, so the maximum growth is at log to the base 10 equals 12. So the max growth is going to be 10 to the 12. And the maximum growth this tumor reaches before it can be detected is about 9. So that's going to be 10 to the 9. So you're going to do 10 to the 9 over 10 to the 12. So the 12, and then you times that by 100, which gives you 0.1%. Figure 7 can also be used to calculate the age of this type of tumor. At diagnosis, a patient had a tumor of 3.98 times 10 to the 11 cells. It says calculate the age of the tumor. So we need to do log 10 of this, and that'll give us what we want. So if we log 10, that, 3.98 times 10 to the 11, we get 11.6. So we need to find where 11.6 is on the scale. So 2, 4, 6, read across, and then down, and you should get about 5 years. Trexol is a drug that can be used to slow the development of various forms of cancer, also known as methotrexate. It's used in a lot of inflammatory diseases as well. Yeah, anyway, Trexol slows cell division by interacting with an enzyme called dihydrofolate reductase, DR. DR is involved in making nucleotides, and the substrate for DR is folic acid. So figure 8 shows the structure of Trexol, figure 9 shows the structure of folic acid. So now it says, so use the information to suggest how it slows cell division. Well, it acts as a competitive inhibitor, as it is similar in shape to folic acid. It binds to the active site of dihydrofolate reductase, so DR, needs to be specific in these examples. And you cannot say they have same shapes or that the same shape to the active site. You need to say it's a similar shape. So therefore you get, less folic acid enzyme substrate complexes. And therefore you get fewer nucleotides for DNA replication. You need to talk about what the actual consequence of that is. So a lot of people kind of just jump into competitive active inhibitor, binds to active site, less substrate, and then they don't say what the consequences are. You need to say what that does and how it slows cell division. So if you can't do DNA replication, you can't do cell division. Doctors investigated the concentration of Trexol given to patients affected by the growth of lung tumors. They measured the volume of lung tumors at the beginning of the study and after 8 months. The results are shown as figure 10, and the bars represent plus or minus 2 standard deviations. A value of plus or minus 2 standard deviations from the means includes over 95% of the data. So here's what we've got going. We've got the concentration given per week and the mean change in tumor volume. So you can see that the volume, the volume is obviously increasing because it's positive. At the point where the line is 0, there's no mean change volume, so the tumor isn't going getting bigger or smaller. And if it's negative, it means it's getting smaller. And you've got your standard deviation bars as well, and all of that kind of jazz. So now it says the scientists measured the percentage change in tumor volume, suggest why they recorded the percentage change and the tumor volume. Well, it's because tumors have different sizes and shapes. So it's obviously, it allows comparison. So it allows comparison. You need to say why. Because tumors vary in size, well, volume and shape, I should say. Sorry, volume and shape. Because if they're what they're doing is they're taking, like, measuring the tumors at the start, and people are going to have differing levels of tumor. They're going to have more tumor, and some are going to have less. So that's the percentage change. The tumor volume is because tumors are different in shape or volume. Because not everyone's going to have the perfectly same tumors at the same time. So recording sort of, tumor volume and shape allows them to make those comparisons and say, oh, yours has gotten bigger or smaller, not yours is this and yours is that. Because let's say if I use like a scale of 1 to 10 and somebody comes and they got a tumor of 1 and I'm like, your tumor's gone from 1 to 5, and then someone else started at 6, it doesn't really allow for very great comparison because 6 to 7 isn't as massive as a jump as 1 to 5. So allowing percentage change puts them all on the same scale. Patient received 15 milligrams of Trexol per week. After treatment, the diameter of his tumor was 35.8 millimeters. As tumor was spherical, use the mean percentage change in tumor volume shown in figure 10 to calculate the volume of the tumor before treatment with Trexol. Let's just get all the pieces together. So the diameter of his lung tumor was 35.8 millimeters. So if you half that, I'll give you the radius, which is 17.9. So half that. Now, this mean percentage change is in tumor volume, so we need to find the volume of his tumor after. So the volume after is going to be 4 over 3 times that cubed. So 4 over 3 times pi times the radius cubed, 17.9 cubed, gives you 24024 millimeters cubed. So you've got the volume of the tumor after. Now, the mean percentage change in volume was minus 25%. So this is 75% of the original. So to work out the original, all you do is you do 24024 divided by 0.75, and that gives you 320. So now it says, to reduce the size of tumors, would it be better to use 30 milligrams of Trexol a week or 20, and explain your answer? So you're going to have to use this good old diagram. So again, for and against, it's an evaluate thing. So if we look at 30, comparing it to 20, so comparing here and here, there's no overlaps in their standard deviation, so there's a significantly greater reduction. So you can say that, and again, you can't just say the results are significant, you need to say there is a significantly bigger reduction in tumor size. If they overlapped, then there wouldn't be. So there's a significantly greater reduction in tumor volume. And in some cases, I mean, it's minus 100. In some cases, the whole tumor's gone. Obviously, we need to talk about converse points as well, like against points. So there's too much variation. I mean, you can see the spread of results is massive. Some people are only getting like, a minus 50% decrease, whereas others are 100. But although obviously, 20 isn't as good, people are getting about 70 to 80 or whatever. Sorry, not 70 to. So people are getting about 30 to 40. At least it's more the range at which people are getting good results are narrower. And obviously, if you're using a fat dose as well, there's probably going to be side effects. Trexol can also be used to slow the development of rheumatoid arthritis, a pain-causing joint disease. Scientists investigated the effectiveness of Trexall as a pain relief treatment in 12 rheumatoid arthritis patients. All of them were female. They randomly divided them into two groups. So Group R got Trexall tablets for 35 days. Group S didn't. The control. They asked both groups to rate their pain on a scale of 0 to 10, and at the start and then every 7 days for 35 days, they calculated the mean scores. So the number of days of treatment in Group R, you could see that the um, the pain decreased, and in Group S, the pain kind of decreased, but it's similar. So apart from age and general health, what are the things you want to consider? Well, obviously, we need to think about other variables that can affect them. So, do they take any other medications? They might be taking pain relief because they're measuring pain. If they're taking pain relief, then their pain's probably not going to be the same as somebody who isn't. As well as that, you need to think about other things like how long or severity, sorry, of their rheumatoid arthritis. Because if they've got really, really severe arthritis, they're probably going to start off with like a really high level of pain. So it's obviously that. Other things we could think about are ethnicity, body mass as well. A student analyzed Table 3 and concluded that Trixol was effective in reducing pain in arthritis patients. Evaluate the conclusion. So again, for and against. So for, the pain decreased more in R than S. But what other things can we say? What other problems are there with this? Well, I mean, they've only tested 12 patients because it's only a small sample size. Also, pain is subjective. Like, I'm a little bitch when it comes to pain. So if I like stub my toe or something, I'll be sobbing and crying. Whereas I know a lot of people who just don't care. They're just like, oh, stub my toe. So that's obviously another tricky part. As well as that, we have no statistical testing. We need to say why. So please do not just say no stats test because you'll get 0 marks. You say there's no statistical test. So we don't know if the difference in pain is significant. You cannot, and I repeat, cannot, please, you just cannot say we don't know if the results are significant. The mark scheme, every mark scheme on the planet says ignore, do not know if results are significant. You need to say what the results are. Now, depending on when I've uploaded this, either I'm going to upload a video berating all of you on this, or I already have uploaded a video and you should go watch it. I can't remember which one's coming first. But yeah. And now here we are at the dreaded essay. Oh no! Right. Don't panic. And you should spend no more than 45 minutes on this. I think, to be honest, you could probably get away with like half an hour. But regardless, all you got to do is talk about maybe 4 or 5 topics and just that's it. You got to talk about, the normal things about them, so how it works, and then the effect it has. So what I recommend is when revising, do one of these obviously when you're practicing the mock, but write the other essay anyway, because like it just helps you practice what you're doing. So it says, let's look at, I'm going to look at the second one because like who the hell would choose the first one over the second one? Let's be real here. But yeah, I'll discuss the other one as well. So the importance of bonding and bonds in organisms. So that's like pretty much the entirety of biology. So think in order of the topics you've done. So topic 1 and you start off with like biological molecules, then you move into exchange and transport systems, digestion, immune system, diversity, respiration, photosynthesis, homeostasis, nervous responses, muscle contraction, and gene tech. So you need to think about all of those, and then you think, okay, what topics are really important? So bonds and bonding, you should be thinking about things like monomers, polymers, carbohydrates, lipids, proteins, and then you could talk about their importance. So for example, the importance of glycosidic bonding in glycogen allows it to act as a store for energy, an energy store. So if you can store energy, that means you can release it when you're exercising or whatever, and release ATP, and you can provide that energy for movement. That's an important thing. That's the importance. You've got you've got things like hemoglobin, so the structure and bonding of that. So you've got polypeptide chains linked together via the primary, secondary, tertiary structure, and all that's a quaternary structure. That's important to get blood and oxygen around the body to allow respiration and energy supply as well. Photosynthesis and respiration, those are very important. Control of muscles and nervous transmission, so but things binding to receptors, neurotransmitters, and stuff like that. You should talk about sort of how they work normally, and then say this is important because if this wasn't here, then this is a problem. So, for example, with the example of glycogen that I mentioned there, if you can't store glycogen as an energy source, you'll have a lot less energy, and if you go through periods of starvation and stuff as well, you're doomed. Like you're gone. You'd have to continually keep feeding yourself glucose, which is impractical. So you need to talk about the importance of it like that. If you stick to 4 or 5 topics that are relevant, then it's fine. I mean, you could like you can obviously try and aim for those extra 5 marks. However, if you risk, if you say something inappropriate, then you risk yourself losing marks and being locked in about like what is it, like 16 marks ish off the top of my head. So if you've got some unrelated topic, you're stuck there. So it's really important just to focus on trying to get the A level content first in. And if you really are feeling ballsy, or go natty in general, then by all means try and go for it. But I've also discussed these essay tips in another video. You should go watch that one as well. I'll link it. Thank you very much for watching. I hope you're doing well, and sorry if I sounded snotty during this one. I'm a bit sick.